Okay, so everybody, welcome join today's talk. My name is Xiaorong. I'm the host for today. I'm currently a postdoc at the University of California, Santa Cruz. So we are super glad that we invited Zheng Dong to give us a talk. We actually met each other at the Golden Conference, and we both we registered super late, so we don't have on-site housing, so I have to drive outside and then come back every day. And he has a poster introduce his work, and it's a really good work. So I'm like, how about you come here to give us a presentation? And he's like, yes, without any hesitation. So now we invited him here. Before he starts, I want to introduce some background of our speaker today. So Zheng Dong got her bachelor degree in bioengineering at Central South University. And then he come to Shanghai to become a PhD student, belongs to both Institute of Neuroscience, Chinese Academy of Science, and Shanghai Tech University. And during his five-year PhD period, he published a lot of work, which I'm surprised because one of the paper published in 2017 has been cited over 200 times. That's an amazing number. And he said his goal of the research is to understand the intersection of metabolic dysfunction and mental states and their contributions to metabolic psychiatric disorders. So his PhD work is basically focused on eating disorders and other things. After he graduated, he came to the United States to become a postdoc at the Boston Children's Hospital with the mentor, Dr. Yi Zhang. And he started to study the addiction. And then he published a really nice work this year. So today he's gonna talk us mainly about the postdoc work. So, okay, so I think the introduction is here. So let's welcome Zheng Dong for the presentation.
Okay, thanks. Let me start sharing.
Yeah, you can start to share.
Okay. Can you guys see the slide?
Yes, perfect.
Okay. Hello, everyone. Thank you for being here for today's talk. And I would like to especially thank Dr. Ma Xiaorong for her invitation and a nice introduction. So today, I'm going to talk about the recent work that we find a specific cell type in nucleus accumbens that can regulate cocaine addiction. So we know that drug addiction has caused enormous burden to addicted individuals and society. For example, in United States, we can see the data on the left panel from CDC. So in the past few years, in every year, the national-wide drug-involved overdose-induced deaths is suppressed more than 100,000 people. And also especially there is a surge increase in the past few years. And the situation in China is much better actually, but still we can see data from the China National Narcotic Control Community, and we can see in the past year, 2021, there are still around 1.5 million people that are registered as a drug user. So scientists has been work many years to find the important neural substrate that control drug addiction in order to implement better treatment strategies. And among those important brain regions, one of the striatum region, nucleus accumbens, which is actually the ventral part of the striatum. So nucleus accumbens integrate multiplex functions of drug addiction, such as reward processing and anticipatory motivation. So it's basically involved many aspects of drug addiction. And from the diagrams we can see here, the NSE is an integrated hub that collect many regions of the brain reward system. For example, it's received dopamine innervation from ventral tegment area, and also receive glutamatergic input from many cortical and subcortical regions like prefrontal cortex, thalamus, and hippocampus. And it's also received like GABAergic input from like hypothalamus, or ventral paradigm or brainstem. So in general, the NSE is an integrated hub. And if we look at the key cell types in the NSE in the past years, in the past, people has been mainly focused on a few key cell types in nucleus accumbens. For example, if we look at the cells in the NSE, the most dominant cells are medium spanning neurons, MSN. So medium spanning neurons is a projection neuron in NSE. It can innervate very far regions, and those medium spanning neurons occupy about like 95% of the NSE neurons. And people also further divide those cells into two main cell types. One cell type expresses dopamine receptor DRD1, and it's a component of direct pathway. And the other half of them, which is actually about like 47.5% of them, express dopamine receptor DRD2, which is a component of indirect pathway. And of course, there are like 5% of the NSE neurons that are interneurons, which has usually innervate nearby neurons. And of course, those interneurons can be further divided based on their molecular compositions. So if we look at the brain region, NSE is a very big region. It's like volume-wise, it's a very big region. And of course, the DRD1, DRD2, it's a very, very large populations. So it's because the NSE's function is so diverse, it's so heterogeneous. Those heterogeneous cannot be only found based on like those molecular heterogeneity. So actually, recent years, our group and many other groups, we utilize like single cell sequencing and multiplex RNA image to decode the molecular and cellular heterogeneity of the NSE. For example, our lab previously we used like single cell sequencing. We can see here both DRD1 subtype, mini spanning neuron MSN, and DRD2 subtype MSN can be further divided into more than 20 cell types. And also we use like multiplex RNA spectral imaging. We can also find both the DRD1 and the DRD2 can be further divided into many different cell types. So in general, those comprehensive cell atlas that provide a basement for investigation of different cell types in complex behaviors like drug addiction. So among those cell types, we find one cell type, one specific very interesting cell type that express neuropeptide Tac2+. And we find in mice, those Tac2 expressing neurons is a specific D1 subtype. For example, you can see here in the upper panel, the green DRD1 and the red Tac2, we can see that Tac2 are almost exclusively expressed in DRD1, but not DRD2 here indicated in the white dot DRD2. And the cognitive result, the cognitive suggested that the DRD Tac2 is almost exclusively, it's like almost 95% of them are co-express DRD1. So it's basically a specific D1 MSN subtype. It's occupied around like 10% of them. And also some other groups also find analog in primate. It's Tac3. So basically the Tac3 in primate equals like Tac2 in rodent. And some other groups also find that the Tac3, which equals to like rodent Tac2, is also specific D1 subtype in rhesus monkey. So in general, the Tac2 represents a novel DRD1 subtype. So we next wondering what is the function of those specific cell type place. So we acquired like Tac2 mice to specifically target the Tac2 cells in nucleus accumbens. And we employed chemogenetic tools and optogenetic tools to directly manipulate, to activate or inhibit those Tac2 cells. And we conducted a series of behavioral assays that has been previously involved in NAC, such as social behavior, feeding behavior, locomotion, and anxiety. And we find actually those Tac2 cells are specifically involved in drug reward. So the way we measure drug reward, the assays we measure drug reward is cocaine conditional place preference. So the rationale of this conditional place preference is, for example, you can image a scenario in your daily life. For example, when you go to your lab, you see a lot of experimental devices, equipment, and you see your lab mate, you see your colleagues. However, when you go to your bedroom, you see your roommate and you see the bed. Usually, you probably watch TV or sleep in your bedroom. So basically, those two different places are conditioned with different your behaviors. And also, this is a case in mice. So the cocaine conditional place preference is we have two compartment chambers, and those two chambers are decorated with different texture. Like here, I use different flow texture. One is grid flow, one is bar flow. And we first, mice are naive mice. We first let mice freely explore the two different chambers and establish the basal place preference. And after that, we condition the mice with cocaine treatment. For example, we first confine mice stay in the grid chamber, and we inject the mice with saline. And a few hours later, we again confine mice into the bar floor chamber. And in this case, we inject mice with cocaine. So in this case, after a few rounds of training, mice establish the memory, or when I stay in this chamber, I always receive saline. And when I stay in another chamber, I always receive cocaine, which make me feel very good. It's like when you go to your lab or when you go to your bedroom, you kind of feel differently. So those reward-based, those cocaine reward-based place preference is an assay to measure cocaine reward. So from here, the result comes, we find actually Tac2 neurons can bidirectionally regulate cocaine reward. For example, here we can see when we express a chemogenetic activator, HM3TQ, in the NAC region of the Tac2 neurons. And when we see in the middle, we see time spent in cocaine chamber. In the control group, which express M-cherry, which express M-cherry, we can see that cocaine treatment alone can induce a very strong preference, However, when we put mice in the cocaine chamber, in the meanwhile, we apply single injection, which can activate Tac2 neurons. So basically, one chamber is coupled with cocaine and neuronal activation. Another chamber we coupled with saline and no neuronal activation. And we can find that activation of the Tac2 neuron almost completely abolished cocaine-induced place preference. And from right panel, the CPP score, we subscribe pre-preference from post-preference. So the positive value indicate a reward positive value indicate a reward place preference. We can see after neuronal activation, those this reward induced place preference is largely abolished. And we next wondering if we next wondering if we inhibit those group of cell, what will happen? So to do this, we use like chemogenetic inhibitor HM40I to allow and to apply with like single injection, which can inhibit Tac2 cells. So, and we slightly modify our cocaine CBP test paradigm. So we use like a subthreshold assay. Basically, we use a lower cocaine dose and a fewer like confined time. So in this case, cocaine can only like induce a very mild place preference, as we can see in the control M-cherry group. So when we use a lower cocaine dose, fewer like restraint time, and we can see that cocaine, there's lower dose cocaine can only induce very slightly, is not significant place preference. However, when coupled with neuronal inhibition, this preference is largely increased. So in this case, we can see that when you activate Tac2 cell, it's actually attenuate your cocaine reward. And when you inhibit those cells, it's kind of like promote cocaine reward. So those cocaine CBP assay is like a quick assay to measure cocaine reward. And we next wondering if the Tac2 is involved in more complex cocaine addiction related behaviors. So we used an assay to mimic human drug taking behavior. So we know usually when we give mice drugs, it's like experimental initiated. We investigator inject mice. It's not mice themselves want to take drugs. So we utilize operant drug seeking assay. Here is intravenous self-administration. So first mice, we subject mice to a catheter surgery. Basically, as we can see in the figure A, we implant a catheter into the jugular vein of the mice. So in this case, we can directly infuse drug into the blood, into the mice blood vessel, mice vein. And also in the operant chamber, mice have two different livers. So as we can see here, the two different livers. So in this case, initially mice randomly press those two livers. However, when they press the active liver indicator here, we give mice a light cue, and subsequently we infuse a dose of cocaine. And however, when mice press the inactive liver, there is no cocaine infusion and no night cue. So after a few days of training, mice will voluntarily like press the liver to execute a specific behavior, like press the liver, so to get drug infusion. So here on the right is our specifically training protocol. So first we let mice to exercise to establish those self-initiated voluntary drug taking, and after that we subject mice into several different doses of cocaine, and we measure their cocaine seeking behaviors. So we can see here we use several behavioral paradigms to indicate the cocaine addictive like seeking behavior. So first is liver press. So it indicates how many times mice to press the liver in order to get drug infusion. It's kind of measure of cocaine motivation. And also we also count the percentage of active liver responses, which is liver accuracy, because in previously we introduced that there are two different livers. Active liver is coupled with light cue and drug infusion, and the inactive liver there's no behavior outcome. So in this case, after few days training, mice will mostly press the active liver because he knows that active liver is coupled with cocaine infusion, which makes them feel good. So this is a liver accuracy. And of course, how many times mice get infused, gets drug infusion, and also like cumulative infusion across the 2 hours training session. So as we can see in the gray indicates the control mice, M-cherry express mice, and the green indicates the neuronal activation HM3D mice. We can see when we activate Tac2 neuron, when mice execute the drug-seeking behaviors, it's largely attenuate those drug-seeking behavior with a fewer liver press, lower liver accuracy, and of course fewer drug infusions. And consistently, when we inhibit those Tac2 cells, use HM40I, we can see those inhibition of this group of cell largely enhanced the cocaine seeking behavior with more numbers of liver presses and also more drug infusions. So far we identify a specific cell type of D1 and in rodent, in both rodent and some other groups indicated is also the case in primate. And we find those Tac2 cells, the activation inhibition of those Tac2 cells separately, like lectively positively regulate cocaine reward in cocaine conditioned preference assay. And also we find this, and also it's not only involved in cocaine reward, it's also involved in cocaine voluntary drug seeking, which is a measure of cocaine addiction. And so if we look at the conventional model, we previously described the larger population of the D1 and the D2, they kind of execute opponent functions, which DRD1 is usually regarded to promote locomotion and promote drug taking, and DRD2 is kind of suppress locomotion and drug taking. However, here we can see actually a subtype of them is kind of unconventional, which is negatively regulate drug addiction. And also this is consistent with a lot of recently a few years when people like apply different mice models in the nuclear kinase and dorsal striatum, people always find their different cell types can potentially have different regulatory functions. So next, we're wondering why those group of cells kind of different with the larger D1 population. So to do that, we try to directly monitor the Tac2 neural activity during the addiction related behaviors. So to do that, we again use Tac2 green mice. We use like single cell calcium image. Basically, we express a calcium indicator GCAM6, which is an indicator of the calcium fluctuations and also like a indicator of neuronal activity. And after that, we implant a miniature microscope to visualize individual Tac2 neuronal activity. So from the so here in the figure A is the behavioral assay we used to directly measure Tac2 neural dynamics following cocaine treatment. For example, mice first go through several days of saline injections like habituation and reduce stress of mice handling. And on the test day, we first inject mice with saline, and we recorded pre-injection and post. We recorded a 10-minute time window of pre-injection and post-injection. And the other day, we like treat mice with cocaine, and we again recorded like during pre-injection, and we also recorded the calcium activity during 2 different time window post-injection. Why is like first 10 minute here is like measure acute cocaine response, and another way is measure like almost 1 hour later is measure like decayed phase, because previously ketamic studies, pharmacological ketamic studies already indicated that single drug injection like can last less than like 1 hour. So it's like initial phase. The first 10 minutes like measure acute response, and the last 10 minutes is measure decayed response. So here in the bottom panel, we can see it's a visualize of individual cells is almost is also like post corrected, and also we extract calcium traces and analyze calcium spikes or calcium transit, use standard algorithms, and we find actually there are two different like kind of patterns of neuronal responses. For example, we can see here on the figure B, some of the neurons they shoot a higher frequency of calcium traces or calcium transit after cocaine injection, and also some of the cells shoot from the bottom, as indicated in the blue lines, is reduced is less calcium transit after cocaine injection. So we quantify those response. Here is a simple heat map of the neuronal transit frequency. Basically, is to calculate the frequency of the calcium spikes, and we can see in the saline injection, only relatively small amount of neurons, like around 7 or 8% of them shoot excited or inhibited after your saline treatment. And however, when we treat mice with cocaine and we analyzed pre-injection and the first 10 minutes of post-injection, we can see a slightly larger amount of neurons that are excited by the cocaine treatment from like around 7% to like 40%. And more strikingly, more interestingly, we actually observed 1/3 of them, like around 33% of them, are actually suppressed by cocaine injection. And from here, from figure G, we can see is a cumulative probability of those transient frequencies. So we can see when we compare the black line, which is pre-injection, the red line is post-injection. When we compare saline injection, those two lines is basically the same. There's no much difference. And however, when we compare cocaine injection, so when we compare pre-injection and the first 10 minutes post-injection and another 10 minutes, like almost 1 hour later, we can see that in the red line, as the red line and the black line indicate, there is a left shift of the red line. Basically, that indicates more neurons are actually suppressed. They are shoot smaller transient frequency. Basically, their neural activity is kind of suppressed. And of course, this suppression is gone after the cocaine activity is decayed. And also here we extract those cocaine excited neurons and the cocaine inhibited neurons. We can see those cocaine excited neurons, the number is less, it's like 49 of them, and the cocaine inhibited neurons, the number is much bigger, like 170 of them. And we can see that those cocaine excited neurons, their neuronal activity largely increased and slightly decayed. And those cocaine inhibited neurons, when you inject with cocaine, their neural activity largely decreased and then slightly recovered. So this is a measure. Like Tac2 neural activity after acute cocaine treatment. Basically, we find that Tac2 neural activity is negatively correlated with cocaine injection. And the next, we wondering if Tac2 neural activity, what is their dynamic during cocaine memory expression? So this is like we measure, we slightly modify our cocaine CBP assay, and we also like place miniature microscope into the mice brain to visualize Tac2 neural activity. And so during the preconditioning, mice are again naive. He did not has any association between specific chamber cues with drug treatment. And after that, mice go through several rounds of saline and cocaine conditioning. And after that, basically mice established in which chamber he received cocaine, in which chamber he received saline. So in that case, we again record when mice freely choose the two different chambers. So this is like monitor our activity. This is kind of like we can imagine a scenario like we measuring our own neural activity when we go to the lab or when we go to the bedroom. So it's kind of because we already conditioned with the lab environment and with the bedroom environment. And when we go to lab or go to bedroom, we record our neural activity. So this is the same thing as the mice. So, and here the figure B, we can see that mice do form strong preference into the cocaine-related chambers. And when we analyze calcium transit in figure C, we can see we analyze calcium transit when mice comes from cocaine chamber to saline chamber, and then from saline chamber to cocaine chamber. Basically, he freely travels through the two chambers. And also here the C indicate cocaine, the S indicate saline chamber. We might travel through different chambers. And again, we observed two group of neurons. One group of the neuron, their neural activity is kind of excited when they stay in cocaine chamber, and their neural activity is slightly suppressed in the saline chamber. And the other group, another group of neuron is kind of, we see their neural activity is kind of inhibited when they stay in the cocaine chamber, and their neural activity is activated, it's slightly excited when they stay in the saline chamber. So this is of course from representative mice, and on the bottom part we can see when we average those calcium traces, we can see those two group of cells show different response pattern when mice stay in cocaine chamber or stay in saline chambers. So we next quantify the fraction of those, we quantify the fractions of those different responsive cells. For example, from the figure D, we can see during the preconditioning, because mice are in general naive, he did not have any clues that of specific chambers. In this case, mice showed a relatively small amount of them. Small amounts of the recorded neurons should like activate or inhibit when they stay in cocaine chamber. For example, it's around like 6%. And after conditioning during the post-conditioning session, we observed a larger increase. We actually observed a larger increase of neurons that are actually kind of suppressed when mice stay in cocaine associated chambers, and the slightly decrease of neurons that are excited when they stay in cocaine chambers. And of course, in figure E, we can see we extract those calcium chamber excited neuron or inhibited neuron. So those calcium chamber excited neuron there, it's like 16 of them, only 16. So we can see, of course, when mice stay in cocaine chamber, their neural activity is much increased. And however, we observed a more amount of the neurons. It's like 62 neurons that are actually suppressed when they stay in cocaine chamber. And interestingly, we find those neurons, their neural activity is actually dramatically changed when mice making their decisions to go to saline chamber or cocaine chamber. For example, if in the figure F, when we compare those cocaine chamber excited neurons, when mice move from saline chamber to cocaine chamber, we align the chamber entry as a time 0. We can see those neurons already firing when mice make the decision to enter the specific chambers. And also this is the case of those identified cocaine chamber inhibited neurons. Their neural activity is actually kind of inhibited when they go to the cocaine chamber. So in this case, we find that actually cocaine plus conditioning can modulate Tac2 neural activity that are associated with cocaine reward contexts. So next we're wondering through what downstream target those Tac2 NAC Tac2 neurons may execute their functions. So to do this, we conducted a simple optogenetics facilitated circuit mapping. We inject trilodopsin into NAC region of the Tac2 green mice, and we identified several downstream targets such as ventral pallidum, lateral hypothalamus, and ventral tegment area. And we next did a functional screening. Basically, we inject trilodopsin virus into NAC region, and we implant optic fiber to stimulate different nerve terminals in ventral pallidum, in lateral hypothalamus, and the ventral tegment area. And we found actually only the lateral hypothalamus is a downstream target to mediate Tac2's function in regulate cocaine reward. So in summary, we found that cocaine administration can actually reduce Tac2 neural activity. And also we found that cocaine treatment can actually modulate Tac2 neural activity when mice enter or stay in cocaine associated chambers. And also we find we identified the lateral hypothalamus is one of the downstream target for Tac2 neurons to execute their functions. So this kind of explain why when we manipulate their activity, it can actually selectively regulate cocaine reward. So in summary, we identified a specific cell type of D1 median spinning neuron and in nucleus accumbens, and we provide evidence from different aspects like anatomy and like physiology and like their functional implications. And we utilized like cocaine addiction related behavioral paradigm like conditional preference and voluntary self-administration. And using calcium physiology recording, we found that cocaine injection can actually reduce their activity, and cocaine conditioning also modulates their activity. And through direct functional manipulations, we found that those Tac2 neurons can bidirectionally regulate both cocaine reward and cocaine addiction, cocaine seeking behaviors. So that's basically all of the today's story. Of course, there are many questions that need to be answered. For example, how about the other cell lung Tac2 cell types, D1 or D2? What is their function implication in Tac2 neurons? And also here, or in cocaine addiction. And also here we show that the neural activity of Tac2 and their projections of Tac2 can explain their functional implication. And we're wondering what important molecules are involved. So those are like answers that need to be, those are questions need to be answered for future studies. So finally, I want to thank my mentor, Dr. Yi Zhang, and also my collaborators like Han Xiao. She actually helped to conduct the challenging catheter implantation surgeries, because think about that, the mice vein is very, very tiny and small. And if you want to implant a catheter into the vein and the mice are still healthily moving around, it's kind of challenging, because previously those surgery is relatively easy in large animal like rat, but it's really difficult in mice. So Han Xiao helped me to do that. And also I would like to thank other collaborators like Ren Chao, Yi Qiong, Richard, and Wen Chang, and also some my lab manager Annie. And I also finally want to advertise a little bit for our lab. So we are well funded, and we are well funded lab, and we study like brain cell heterogeneity, use like single cell sequencing and through Murphy's collaborations through Murphy's, and our main focus is like neural epigenetic and neural circuitry mechanism of drug addiction, learning, and memory. And we are actually right now, we're actually actively recruiting postdocs. So if you are interested, you can directly email Dr. Yi Zhang, or you can scan the QR code here. So to look what specifically we are doing. So that's all. I'm happy to take any questions.
Really great talk. So if you have any questions, please use the chat box or just unmute yourself and ask a question.
Yeah. In either way, you can like type or use Chinese or English, whatever. No much to.
你好，赵老师。
你好。
啊，对，我有一个问题，就是说，我觉得您对您的研究非常感兴趣啊，就是说，这个你既然是研究了成瘾，那你用了一些这些范式，比如说这个 CPP， 然后自给药，那我想这些范式都是从一些普通的奖励，你做一些奖赏的效应的时候，它也能做出这种，平时也使用这种范式嘛，但为什么就是说它哪里体现出来成瘾性了呢？就是我想的话，假如说你用糖水的话，是不是你有没有做过类似的实验，就是用糖水记录一下它的 CPP 的那个神经元的反应？
你这是个好问题，就是说，因为 ANC 它就是说，它对很多 reward，like drug reward、nature reward，like 就是这个sucrose可以把它认为是 nature reward， 对吧？就是说，我们没有 direct measure like sucrose， 但是我们 measure那个 food intake， 就是干的食物是没有区别的。
干的食物，那
对，sucrose 我们没有具体量。
OK， 那你记录了它的干的食物的钙成像吗？还是
没有？因为我们 manipulate 它的 activity 并没有明显的，所以我们就没有 follow。
OK。
然后你说的这个 addiction， 这其实是一个很好的问题，因为addiction 就是总体来说是一个非常非常强的一个词，它就是一般 addicted mice， 它其实有点 crazy， 它为了这个吸 drug， 它都不 care， 它都不 care punishment， 不 care 不 care 惩罚，不 care 其他任何东西。它是一个很强的词。然后我们这里其实主要的 measure 的就是我们没有 measure 的如此之强的这个，我们没有用这个非常强的这个 assay， 我们主要是 measure 它 self initiate， 就是 voluntary 的 drug seeking。但是我们没有 measure 它 compulsive， 就是强迫性的，不吸不行的那种。
但是我还是有一个问题啊，就是说，你看现在，比如说我们不管是观察它，就是它我们就像你说的，它强迫也好，或者是我们就算按它 CPP 也好，但是这些范式的话，我感觉你用糖水做跟用毒品做好像没多大行为上的区别。
还是行为上的还是有一些区别的。糖水一般不会有 compulsive。
糖水为什么不会？糖水你用糖水，我不知道你了解过没有？如果用糖水的话，电击的话，它会比毒品更强烈，就它能忍受更大的电流。
糖水，但是他们的 mechanism 是有点不一样的。我给你举个例子，比如说你，比如说你，它可能没有那种叫 sensitization。糖水就是 drug treatment， 它有的时候会有 sensitization， 就是它的这个 locomotion 啊，它是越来越强。但是这个糖水它可能是没有的。然后他们的 mechanism 可能是有点不一样，因为这个 cocaine 它是 directly abolish 的 dopamine reuptake， 就是它直接就是干扰了 dopamine 的 reuptake， 让那个 striatum 的 dopamine 的 concentration 就大幅度的增加。但是糖水它应该是 indirect 间接的作用于这个brain reward system。
OK。
然后就是对您说的，其实糖水确实是一个研究的人也挺多的，就是高 palatable food 研究的人也挺多，他们范式是很类似的。
对。然后我最后一个问题，那既然我们用这些范式，比如说我们这些做成瘾的范式，它观察不到 糖税跟毒品的区别，那是不是就要求我们要发展一些更好的范式去捕捉这样毒品所特异性的行为呢？你就认为有没有必要？
有啊 ，sensitization 就是一个比较特异，还有一些 tolerance 啊这些。然后因为这个RBACA 它已经是一个比较standard 的，就是很好的 mimic，就是人类的 drug taking 的人类嗑药的行为。其实相较于其他精神类疾病，其实药物就这种自身给药已经是非常接近人类的。你像其他的，比如说 depression 啊，或者是自闭啊，其实小鼠行为很难很好的 mimic 人类的情况。
OK，我只是觉得对，用这样的范式，去做的话，假如说你不去跟一些自然奖赏物去做比较，可能发现的大多都是一些跟奖赏相关的一些，比如说一群神经元。我只是这样在想，是不是有这样的可能性？
是的，是的，他们垃圾的 overlap， 对的，
对，
是的，
好，
确实是这样的。
OK，谢谢你，我没有问题了，我就这些，谢谢。
好，谢谢，您是做什么呀？
我做成瘾的，是做猴子的成瘾。
哦，I see，猴子更牛逼。
没有，我们是想，我们是在企图，就是我们已经做出来，就是用一种范式的话，就可以去区别正常的奖赏物跟毒品的一个这个区别。
对，我看评论里面有2个其实很好的问题。
OK，那我
对，你那个，我就是稍微岔开一下，就是评论里面我看有2个很好的问题，一个是问 LH NAC target 到 LH 的细胞类型，这个问题很好，但是我们现在没有固定答案，但我推测应该是 GABA 这个地方，但是这只是推测，但是我们没有特别具体做，因为这个需要当然可以在 anatomy 上稍微看一下，比如说你定量一下它的那个 nerve terminal， 就是跟这个 NAC 不同的。跟 LH 不同的细胞类型进行 staining 染色，但这种 anatomy 证据是比较 weak 的。另外一个，你可能就需要电生理，比如说你 cross 这个 Tac2 cream mice with 这个 GABA 或者 glutamate Phenippus 老鼠，然后一方面你去看，你就能够 target 你电生理去直接 target 这些 cell， 然后 direct measure 这个 post synaptic current，但是我们就是没有这个老鼠，因为这个确实是个很好的问题。然后第二个就是 Tac2 神经元释放的 NKB 及其受体的作用，我们其实做了一些 preliminary data， 我们发现其实 lockdown 这个 Tac2 neuropeptide 它可能效果没有很好的效果，可能的原因是因为这个 Tac2 它 express 到就是它大脑里面 express Tac2 的神经元还挺多的，就是很多脑区都有。然后我们觉得就可能 lockdown 一个脑区，因为 neuropeptide 它本来很多时候就是 volume diffusion， 就是它可能不是那种对就特别精确的那种调控，所以我们觉得可能 lockdown 一个脑区可能会有这种补偿。然后所以我们其实至少在这篇工作当中，我们其实强调了它是一个 D1 的一个亚型，我们主要强调它的细胞的作用，没有特别的去强调它 molecular 的作用。然后这个 NKB 的话 ，NKB 的话，它确实是一个Tac2，它因为 Tac2 是基因名字 ，NKB 是它就是转录之后的这个 neuropeptide 的名字。然后 NKB 它其实我们试了一些这个 agonist 和 antagonist， 它的 agonist 它的代谢的表型非常强，这个它会对老鼠的这个体温和它的活动其实影响还蛮大的，所以就很难去 direct measure 它的 drug 的 effect。然后 brain region specific 的，其实我们尝试了一下，其实在有些脑区它是确实在有些脑区它是确实可以影响 drug reward， 但是它的那个副作用非常强。然后 NKB 还是 GABA， 就是说从目前我们从 cell 的角度来说，我们可能认为是 GABA。 比较重要。从细胞的角度，但 NKB 还要进一步的去看 neuropeptide 这个问题，这几个问题都挺好的。您是这个领域的吗？我感觉，
哎，郑总你好，我做这个有几个比较naive的问题，就是我看你那个 GI 和 GQ 那个 dose dependent，好像是1的那个都是没有效果的，是浓度太高了。
对，这其实也是一个挺好的问题，但是我们就是发现这个药物给的太高了之后，它的 neural manipulation 可能就影响就不大了，因为它这个药有点给的稍微高了点，
是它一下子就
你观察的挺仔细的。
是说药的浓度是说就是它一下子就叫什么就怎么说就老鼠一下子快感就达到
应该是这个原因，因为你 neural manipulation 就是你那个神经操控实际上是让老鼠不那么 reward 的，但是如果你给的很强，但是这个 reward 没有被完全 block， 在完全 block 好像似乎也不太 make sense， 就是说你如果用了很高浓度的药。至少我们观察到的是这样。如果用极其高的浓度的话，它没办法就是影响。
还有一个那个 slide 12，就是那个 one photon imaging 的，我就发现你这个cocaine 的那个117个被 inhibited cell， 它的 baseline 负10到0分钟的这个 baseline 好像就是要高一点，
因为它都是 normalized， 它就是你分析 calcium image 的，你实际上是 normalized to entire session， 所以它neuronal 就是你鉴定的 inhibited neuron， 因为你是 normalized 之后的嘛，所以你前面总体来说看起来总会是相对来说兴奋的，就是你给药之后是抑制的，你给药之前那肯定是兴奋，因为你们两个之间是相互比较的，所以它确实会有这种情况。在这种 calcium，因为它 calcium 它是一种相对性，它不是绝对的，不像 electrophysiology 它可能是绝对的。你直接去 measure 它的 volume，
明白你意思，就是你有没有做过更细的分析？就是说，在 saline 情况下被抑制的，在 cocaine 情况下被抑制或者是被兴奋的比例，反过来在 saline 被兴奋，在 cocaine 的时候被兴奋或者抑制的那个。
这个挺好。我们没有，我今天没有秀，但是我们比较了一些，就是在 cocaine 就是早期和晚期的。这个不同 neuron 它的变化，其实发现这个 Tac2 neuron 对这个 cocaine treatment 的这个还是挺 dynamic 的，就是它刚开始它响应，它后面可能它会有一些 overlap， 但可能就没有，它其实有 overlap， 但是这个 overlap 可能40%到50%这样嘛，所以你也很难说它 overlap 就是它的 initial phrase 和 decay phrase 你也很难去就 direct 的就直接下一个结论是一样的或者是不一样的，会有一些overlap。
还有最后一个问题就是，我想确认一下你那个 working model， 就是说 Tac2 它是个 governorgic neuron， 它在 cocaine 的时候被抑制，所以可以 disinhibit VTA 的 dopamine neuron， 所以 VTA dopamine 会释放更多 ，cocaine 可以带来更大的快感。这是你的 working model 吗？
我们是 LH。这里我们发现其实主要的作用是 LH，VTA 本身好像并没有特别明显的作用，因为你说的是一个 feedback，对吧？就实际上 feed forward back 就是ANC 的 Tac2 影响了 VTA， 然后 VTA 又进一步影响了 ANC，对吧？你这说的这个，但是我们其实发现 LH 是它比较重要的项目。可能在这个 context 下，可能 LH 可能比 VTA 更加重要，可能啊。
谢谢。
谢谢。
那个赵指导，我也几个问题想问一下呢。
好呀。
就是第一个就是那个你抑制或者激活，你们看见Ocamps 的 Tac2+本身是个 reward， 或是一个 reward 或者是个 aversive 的一个现象吗？
这是个好问题，就是说 neural activation itself。
对。
我们没有特别直接的去比较。 我们只评价了它对药物 reward 的影响，他们自己直接的影响，我们并没有直接的去，因为它，对我们没有特别直接的去比较。
OK， 另外一个就是你们在测那个就是那个 preference indication 的时候，你们在后面测的时候，你们也是就是给那个 CNO 去激活去什么，就是说在后面就是condition 完之后，在测的时候就没有给那个
没有了，没有了，就相当于你没有了，就是这个 cocaine 就是你可以看这个 paradigm 这里，这个 neuronal activation 和 inhibition 我们只在 condition 的时候，也就是说 condition 不是把它 treat with saline 和 cocaine 嘛，对吧？就是我们这个时候在它给开 cocaine 和 saline 的时候，我们给 CNO 就相当于评价一下它 neuron activity 对直接这个 cocaine reward 的影响。最后 post conditioning 我们是不给药的，就相当于你天天去，就是你已经 condition 到你的实验室和你的寝室，所以但是你平时比如说你某一天去了实验室，某天去了寝室，但其实这个时候你已经 post condition 就不会再给药。
那我就非常好奇，就是如果你们在这个 post 的 condition 是在测的时候，你们就是说，如果在前面我不给药，我就去正常的这个这个险，在condition 完之后，我在measure 的时候，我去就是给 CNO 去或者激活这个 Tac2+neuron，它会对这个
其实你说的这个，其实你提的这点还挺好的，因为你这个问题它涉及到两个事情，就是 memory formation 和 memory expression。
对。
memory formation 就是说你在小鼠 establish 这个 memory 的过程当中给它操作，就影响 neuron activity， 看它对后续的影响，对吧？然后 expression 就是我老鼠已经建立了这个 reward， 已经 condition to 这个 reward， 然后呢，它最后要表达这个 reward， 就表达 reward 就是通过我往左走，往右走，往这个前面走，往那个前面走，然后我在表达的时候给 neuron 刺激。这个时候有不一样，所以你其实这个提出的就是说，我们关注的是这个 reward 的 formation 还是这个 reward 的 expression？ 但是我们这里其实主要关注的是 reward formation， 就是说它在 format 这个它在形成这个 reward 的时候，给它cocaine reward 的时候给它 neuron 的给它神经刺激，看它的反应。
那就是你们有好奇的，就是去测一下，是它的这个expression 它会有了影响吗？
我们没有测，因为这个是个不同的问题。
OK， 然后还有最后一个问题，就是在你的那个 recording 的时候，我看就是它不是就是Tac2+neuron在cocaine它本身的反应的时候，有很多就是就是有些会上升，然后有很多会下降嘛。
对。
然后在后面就是在condition 之后，就它也是有很多会下降，有很多会上升。我想问就是这两群，比如说都在下降的这两群neuron里面，就是对 cocaine 本身有反应，是本身下降和就是在 condition 之后下降的这神经元，他们是一群神经元，还是说他们是不同的？
好问题，就是说我们用了两个不同的 assay， 一个是 cocaine 的acute treatment， 然后一个是 cocaine 的一个condition， 对吧？然后你其实就想问那些在 cocaine acute treatment 响应的神经元，是不是后来演变成了这个在 cocaine condition 过程当中也有反应，对吧？你是这个问题，对
吧？
对，这个是个很好的问题。但是因为我们其实有的这个老鼠它不是完全的这个同批次的老鼠，所以我们没有特别去直接比较。其实这确实是一个很好的问题，其实可以考虑一下。
对，如果它是不同的话，就非常有意思了。
它是不同的话，那就说明就是它的 acute response， 就是它负责它 acute response， 它和它负责 它这个建立 association 的这个 memory 这个过程当中，neuron 可能是不是同一群，那其实确实，但是我猜应该应该是垃圾的 overlap， 肯定会有不一样，但是我觉得可能会有 overlap 会比较多。
OK，
幸好你不是 review 啊。我们其实当时也挺担心 review 问这个问题，因为这样的话你就要补很多实验。
对，这个比较就太麻烦了，太难做了。
而且最主要的是，就是说这个问题它最难的地方在哪呢？就是说因为你这个实验是不同时间做的，你相当于你后面做的你要 align 到前面，但是你想一想，你这个你每次你隔天做你的这个 lens placement， 就是说你要完全 align 好的话，其实可能对大家会这样做的，但是其实可能会需要一些复杂的计算，就是稍微要麻烦一点。但是确实会有人这样做的，可能在突风岛里面大家做的更多一点，因为它记录的更就是它的 neuron 就是记录的更清楚 ，resolution 更高。像这种叫宽场的，它有的时候 neuron 之间 overlap， 就是你其实有的时候记录的一个，它可能不在一个 level 上面，这个时候你把2次不同的记录去 align， 其实就会比较困难。
是的，跟人讲，好的，
你们
谢谢。你说，
你们问的问题都挺好的，谢谢。
郑东你好，我有一个非常傻的问题，就是这个
没有
cocaine 它不能够直接注射到脑子里是吧？
可以啊，
就是一定要，就是因为你说那个就是静脉注射这个手术非常难做，为什么不能够直接 ICV 就是脑室注射？不是很简，那个会很简单。
他这个是明白你的意思。
我
脑室注射就直接注射到脑室，它有一个问题就是因为你注射到静脉里面，你是可以多次注射的，而且这个是 mimic 人的行为。你注射到脑室里面，你想你能注射多少次呢？那老鼠如果注射个10次，它脑子是不是得有点胀？就是可能 technical 上可能会，就是说手术上会简单一点，但是你真正给药的时候就会比较 tricky。但是因为 cocaine 是作用于中枢的，它是直接作用于这个中枢里面去 block 这 cocaine 的block 这 dopamine 的 reuptake， 所以你直接打到脑子里面肯定是可以的，没有问题，这个肯定没有问题。但是我在想你比如说老鼠 press 的10次，你要 infuse 10次，那是不是一下子它脑子有点胀？
很多做手术至少1两微升的老鼠都是可以的，可以 handle 1两微升的。
对，我们打药，因为我也往中枢打过药，但是我主要是往脑区，我没有往那个脑室。我一般用0.3微，不对，0.3微，对。
你这个往静脉就是每次打多少呀？就是我有个概念。
0.03毫升，就30微。
那很多。好，
不多，静脉还行。老鼠你往它静脉里面打，
还行，是。
你往它静脉里面打，我觉得打1毫升，应该可能问题都不大。
好，谢谢。
我的第一个问题跟之前魏超和尹如平问的那个 Tac2 神经元如何编码 cocaine rewards 会有相关性。当然，你之前也回答了一些，就是，我觉得你数据应该有，就是你注册过很多次 cocaine， 我想知道，比如说第一次被激活的，是不是一直都会被激活？被抑制的，是不是一直都会被抑制？就是在你打 cocaine 的时候做钙成像，然后看 Tac2 那群神经元，然后比如说你第一次打 cocaine 它被激活，你第2次打的时候它是不是也是被激活？第3次打是不是也一样被激活？
明白你的意思，就是说，因为我们一般我们就是 single， 你这样说我们没有连续的，就是你的意思就比如说你打一针，然后过1个小时再打一针，再过2个小时或者这种再打一针，看它就是 repeated， 它是会 repeated suppress Tac2 还是其他的，但我们没有去。
对。
我觉得应该可能会有 overlap，因为每次有30%。然后
是。对，然后这个问题就跟我下一个问题有关，就是 Tac2 神经元是不是这是个比较广泛的问题？就是你觉得 Tac2 神经元是不是最小的一群？你觉得 Tac2 神经元能不能找出另外一群？就比如说，假设你将来有转基因工具，只找出来被抑制的那一群。
因为这个永远无法分下去，因为你是根据它的分子构成来进行分的，你就跟着它的表达的分子就是 cell type， 它这个永远没有止境的，因为你 Tac2 肯定后面还可以再分，即使 Tac2 它neuron可能只有10000个，但是你仍然还可以再分。
对，然后另外一个
实际上我们其实就是那个 single cell sequencing 里面，其实我今天没有秀那个就是那个 single cell sequencing 那个图，它其实是有一些，它这个 Tac2 它不是 exactly 正好那一小群，它实际上稍微有一点分为2个小群。
对，然后最后一个跟实验设计相关的，你一开始为什么会选 Tac2 呢？就像你说的，比如说我做 single cell 发现了100个 marker， 我应该怎么选？
有多个原因，就是第一个它当时有 available 的老鼠，第二个其实我们之前测过一些就是 single cell sequencing， 我们跟 cocaine cover 它这个，但是因为当时这个细胞数量非常少，因为这个 Tac2 它neuron也不多，所以那个数据不是很 solid 的。但基本上这个 Tac2 它基因变化还是挺多的，但是那个数据因为它是基于几十个neuron，所以我们就没有把那个数据专门去弄。但是这个 Tac2 第一个它就是比较 specific， 第二个就是当时有 available 的老鼠，第三个就是我们一些就是比较粗略的 data 发现它还是有一些基因变化的。在 cocaine treat 之后。
就是说 cocaine treat 了之后，比如说 Tac2 的表达会上升或者下降。
不是 Tac2 倒没有，因为它这个我们倒没有直接观察到 Tac2 基因本身的变化，当然我们其实观察到一些receptor 的变化 ，GPCR 其实，但是因为它细胞数目比较少，所以就这个结论，因为如果你基于这几十个细胞去下结论的话，可能你也很难往文章里写。
明白，就从实践上来说，至少在小鼠里，比如说我的这群细胞大概有10000个，就已经算是比较小群了，对吧
？10000个，10000个算正常吧？我记得 A 加 P 是5000个，PAM-C 是8000个，Tac2 多少个这个也不知道，但是我觉得可能也在5000左右吧，5000到10000之间，整个neuron里面。那整个 ANC 那就大了，D1 那就大了，那
明白。
好像有人举手，周
对，我就是挺好的，讲的非常清楚 talk， 我就有点疑问，就是说你这个 Tac2 的话，就是你这个基因它就是在你给 cocaine 的时候，有一部分是抑制的，就是同一个表达这个基因的，有部分抑制，有部分是那个就是 exciting 的。那这是因为它们投射的位置区域不一样呢，还是就是你有没有找到这2个差别在哪儿？
不知道，你也没法知道，就是因为这个有一个技术原因，就是说，首先这个 calcium image 它是 association， 它这个 physiology 它是相关性，就是说你只能证明你这个 neuron activity 跟它那个相关，不能因果。也就是说，你相关性你就肯定有激活的有抑制，你很难，无论说你这个 neuron 多么 specific， 多么特异性到你的方式上，你总会观察到一部分上升，一部分下降，这是没有办法的，因为它是一个相关性的 study， 就像你 GWAS 一样，你 GWAS 鉴定的就是你那个 genome wide association study， 你 GWAS 了之后，你发现一个基因跟你的疾病有关，那你能说这个基因就一定有相关性吗？其实不一定，它是一个相关性，因此这是一个，另外一个，它有没有可能是因为细胞组成的不同或者 projection 的不同，这个需要的，就是需要进一步来看，很难在这个 calcium image 上直接看出来，这是没办法的。
对，我就看你后面不是做了那个 optogenetic 嘛？你说就只是看就是这个它投射到 lateral hypothalamus，但是会不会就说你这两个一个 inhibition 和 exciter 它那个投的就不一样？你有没有办法就是通过就是比如说 optogenetic 再 combine 在你的那个什么 calcium image 来去找到这个投射的差异啊或什么的？
技术上挺难的。比如说你可以，比如说你可以在 LH、在 VTA、在 VP 打 retrograde
对
virus，然后在 ANC 记录，然后你再比较它的这个比例。但是这个实验就是也是一个挺大量的实验。
对对，就是
尤其是你 calcium image，你本来 image 这个 cell type 就挺 tricky 的，然后你现在又加了一个 retrograde tracing， 它细胞数目可能会不多，所以你记录起来可能会有点，可以通过这样，但是 anyway， 就是它有一个技术问题永远无法克服，就是它是一个相关性，就是无论你怎么去 define 它总归会有一部分上升，一部分下降，一部分不变，这个是永远没办法改变。
就是像他们
但
刚才
是它的比例
是不是它永远都是这个这群 exciter， 就是永远都是你给 cocaine 它是 exciter， 然后那个 inhibitor 永远都是 inhibition 的。就是如果是你能看到这个，
就是说不是，就我的意思是这样的，就是说你在就是说你记录一个 calcium image 跟一个 behavior 的反应，对吧？那你要不激活，要不抑制，要不没有反应，对吧？可能根据你不同的 criteria， 就是那个你不同的标准。但是无论你怎么去精细，这三群你不可能记录到一群 neuron 在 calcium image 里面，你记录到一群 neuron 只被一个行为兴奋，这是不可能的，因为它是一个相关性。
哎，
但是它的比例会不一样。就比如说你记录 Tac2， 你整个记录 Tac2， 你可能会观察到1/3，对吧？然后你如果记录 LH 到 Tac2， 那你可能记录到了1/2或者是2/3，这个时候就说明你这个就更加特异了。但是你只能永远的去从1/3到2/3，然后到3/4到4/5，你永远不可能达到。你从来不会在任何一个 calcium image 上面看到一个100%，我不信，这是不可能的。你们它会有很高，比如说90多，这已经是极其完美的情况。
它只能说一个相关性，不能说明就说这个，好吧？
所以就是你要辅之以这个，就是你的 physiology calcium recording 的话，你要辅之以这个 manipulation，就两者之间相互辅助。
谢谢。
我顺着周金师姐这个问题往下再问下来，就是说你看你这里就是那个15那地方，然后你这里不是投到 VP 的这个地方，就是感觉是很多的，是吧？
对。
然后就是你激活就是在 VP 就这个 terminal 是没用的，然后你激活就是到 LH 的 terminal 是用的，那就是有没有可能就是你在microneal complex 里面是对可卡因有反应，或者说对可卡因这个就是
没反应的都在 VP 是吧？因为
他没反应的都是投到 VP 投到 VTA 的，他有反应的都投到 LH 的。
这个其实说实话，我个人觉得这个可能性比较小。这个世界上没有这么完美的事，就是说，
就是它没这么完美，但是就像你之前
有可能，
我那个比例就瞬间就比如说有反应的，不管是一个三十还是inhibitor反应的，累积起来可能就到了90%以上了，就这样子的啊。
对，但是你要是就是说你的 terminal 有好几个，然后你的 neural response 有好几类，但是彼此之间的是不是有直接的对应关系？这个需要那个啥，这个有点难。
然后还有个问题是，我想知道就是，在 neural competence 是这面这些 neuron 的话，它是一个 neuron 投到一个脑区，还是说一个 neuron 可以投到多个脑区去？
一个 neuron 可以投到多个脑区，一个 neuron 是吧？应该可以投到多个脑区。
我觉得这个是之前已经有那个。
这个我没有太关注，就是说这个做起来就比较，这个我不知道有没有人直接去做过，但是一般来说应该可能是一个 neuron 可以投射到不同的脑区的，它的 branch 可能会那个，但这个你做起来就比较复杂，你可能你要证明这一点的话，你就得 spice labeling， 然后
没有你直接在那个比如说在 VP 或者 LH， 就是在那个不同的 CTD， 然后你在 NSA 然后去看他们有没有那个
就是说你
这个就是比较粗嘛。
对，
大概就是
可以的，可以，就是你就是想看，就是它投射到不同脑区是同一群还是不同的群？
对，
对，我看评论里面有人说记得马克安德曼报告说过 ，MSA G4 MC4 neuron 是1/6，然后有一个随机过程。对，就是说 neuron science 里面它有好多是，或者说生物里面的有好多是相关性研究，相关性研究就你怎么做都无法做到100%，它也不可能不 make sense。是的。那行吧，谢谢各位。我觉得
非常感谢。
我觉得今天有好多人的问题挺那个啊，幸好review没问这些前人节的问题，要不然我这又得 delay 了好长时间。
好的，那我们今天就到这儿，非常感谢你来跟我们分享。
谢谢邀请。
对，
挺好。
然后还有后面的那个很好的解答，然后希望大家可以关注我们下个月的报告。好，那我们今天就到这里，谢谢大家。
谢谢大家。